This is our presentation on the effects on disease vectors and the indirect impacts of climate change on human health. In this presentation, we will be covering an overview on climate change, disease vectors, vector-transmitted diseases, malaria and the Anopheles mosquito, past mitigation strategies, as well as current research endeavors, and finally our references. Climate change is when there is an extreme change in temperatures and weather patterns. The main cause of climate change is human activities such as deforestation and the burning of fossil fuels, which releases heat-trapping greenhouse gases into our atmosphere. The effects that climate change has on our environment include rising temperatures and sea levels, and adverse impacts on the ecosystem and human health.
Climate indirectly impacts the spread of vector-borne diseases. A vector of disease is defined by the ESCL as a living organism that transmits an infectious disease agent from an infected animal to a human or another animal. These kind of diseases are transmitted by fleas, ticks, and mosquitoes carrying pathogens that cause illness. Climate change can lead to vector and pathogen adaptations, causing shifts or expansions in their geographic ranges. Geographic shifts can alter disease incidence depending on vector-host interaction, host immunity, and pathogen evolution. Ticks and mosquitoes are common vectors we frequently come into contact with in Minnesota. Vector-transmitted diseases, or vector-borne diseases, are illnesses being most common in tropical and subtropical regions of the world. These insects carry pathogens such as Lyme disease, West Nile virus, malaria, yellow fever, etc., and they transfer these bacteria, viruses, and parasites to humans through bites or other forms of contact with the vector. The return of malaria has raised a lot of concern in North Americans. Originating from Africa, the Plasmodium parasite is spread through the Anopheles mosquito after taking a blood meal from an infected host. The parasite, after 10 to 21 days, is now found in the mosquito's salivary glands and ready to infect the host of its next blood meal. The recent spikes in temperature combined with urbanization leads to new breeding grounds for malaria.
Here we see a brief timeline on some of the methods of malaria treatment over the years. Quinine was the first and is derived from the cinchona tree. It was first used by the native people of Peru as a remedy for common ailments. Chloroquine was synthesized out of necessity for a substitute of quinine. Mefloquine was shown to be effective against the falciparum malaria, although use was discontinued as mosquitoes began to build an immunity and tolerance. Artemisinin are still well established and used today. It has a long history in Chinese medicine and is derived from sweet wormwood. This slide shows the source materials in which quinine and artemisinin are synthesized from. So it is cinchona bark from the cinchona tree and sweet wormwood. DDT was one of the first modern synthetic insecticides in the 1940s. It was initially used as a tool to combat the spread of vector-borne diseases like typhus and malaria, and was later found to be effective for general insect control for crops, livestock production, and gardens. Only 30 years later, DDT would have an EPA-issued cancellation order based on observed adverse environmental effects and potential health risks. Research is still being done to determine whether DDT is capable of causing reproductive problems or birth defects in humans. However, animals such as rats, mice, and birds have shown to suffer from these upon being exposed to DDT. The most familiar example would be DDT's effects on the bald eagle population that resulted in a decline in reproductive success and thinning eggshells. The image shown is an illustration that appeared in a color print magazine advertisement for Pensalt DDT products, which appeared in Time magazine June 30th, 1947. Contrary to the text, DDT is not in fact good for you. Transgenic mosquitoes are a popular and controversial topic when it comes to the research being done to control specifically Aedes aegypti mosquito populations. These populations of genetically modified mosquitoes do not work to control the population between species of mosquitoes, meaning genetically modified Aedes aegypti mosquitoes will only work to control the population of other Aedes aegypti mosquitoes. Genetically modified mosquitoes have shown to be successful in the control of mosquito populations when released in parts of Brazil, the Cayman Islands, Panama, and India. When genetically modified mosquitoes stop being released, the mosquito population will slowly even back out to normal levels. Insecticides are drugs commonly used in livestock production to control parasites. Insecticides, specifically ivermectin, is also being looked at as a possible mitigation method. Though experts warn that just like any insecticide, it's possible that mosquitoes could build a tolerance or immunity. Research is still ongoing, but ivermectin has been commonly used to treat other vector borne diseases such as onchocerciasis and elephantiasis. The image shown in the slide shows a lab technician displaying Aedes aegypti mosquitoes infected with Wolbachia bacteria, which has shown great promise in blocking the replication of viruses in the midgut of the mosquito. In conclusion, climate change is only exacerbating the spread of vector borne diseases. Despite this, experts are constantly researching mitigation methods, and notable methods such as the use of Wolbachia, insecticides, and genetic modification technology have shown great promise towards lessening the transmission of vector borne diseases.